Good afternoon. Just to say, we'll be starting at the top of the hour in about a minute. So welcome to the webinar today. And in the meantime, please take the time to fill out the demographic questions that are appearing here in a poll on the screen. If you're just joining us, we'll begin very shortly. Please take a moment to fill out the demographic questions on the screen. So I guess we should begin today's session. It's 1 minute after the hour. So I just want to welcome you today for the webinar on an overview of data presented at the 2024 International AIDS Conference. It's my great pleasure to be here today with Dr. Paul Sachs. I'm Marina Klein. I'm based at the McGill University Health Center at McGill in Montreal. And I think Dr. Sachs is well known to most of you. And I'll introduce him a little bit more in a moment. So just before we start the presentation today, I just want to go through a few introductory slides. These are the disclosures of the content on the web board listed here. These are also the disclosures of the presenter, Dr. Sachs, and myself, as well as the reviewers of this material. Just to remind you that this is an IAS-USA designates this live activity for a maximum of 1.25 CME credits. It's also a webinar approved for 1.25 hours of ABIM MOC points and nursing, pharmacy, and pharmacotherapy credits. The generous support for this activity has been received from the following contributors. And this is an important slide because this is how you can navigate this activity. So there will be poll questions like there was at the beginning that will pop up in a separate window. Please choose your response to the poll, and then we'll display those results after the poll closes. When it comes to questions, which we really encourage you to submit, use the Q&A button exclusively. So we may not be able to address all the questions that are there, but we'll definitely be monitoring the Q&A. The chat is open, but this is not the place to submit the questions. So now it's my great pleasure to introduce our speaker today, Dr. Paul Sachs, who's the clinical director of the Division of Infectious Disease at Brigham and Women's Hospital and a professor of medicine at Harvard Medical School. And he holds numerous important positions and editorial positions, including with clinical infectious diseases. And he teaches regularly on HIV and infectious diseases locally, nationally, internationally. And in addition to being a superb physician and teacher, he's also a researcher who works on clinical trials of antiretroviral therapies, cost effectiveness of management strategies for HIV, and toxicity of antiretroviral therapy. And so I'm going to turn it over to you, Dr. Sachs. We're looking very much forward to the presentation.
Thank you, Dr. Klein. It's an honor to be here. I'm going to show my disclosures again. And now we're going to go through the learning objectives, and I'm going to make fun of them as I normally do, which is not something an educator is supposed to do. But in fact, we are required to have learning objectives. And so we're going to go through what we think are the most important data from prevention and treatment and complications at AIDS 2024, list potential applications of the data to clinical practice and incorporate relevant research into clinical practice. But first, as usual, we're going to challenge you with some pre-test questions. And so here comes pre-test question number 1. in the study of lenacapavir and tenofovir alafenamide and tricytidine for pre-exposure prophylaxis in women, which of the following was observed? A, breakthrough infections leading to resistance to lenacapavir. B, superiority of TAF-FTC over background incidence of HIV. C, progressively worse adherence over time in the TAF-FTC arms associated with risk of acquiring HIV. Or D, significantly higher incidence of HIV in the TAF-FTC group than the tenofovir diazoxyl fumarate group. Okay, you're going to go ahead and vote. I'm not going to give you the answers, not yet at least, and we'll ask you the answers again at the end. I'm just going to set this little quiz aside. And Dr. Klein, you want to be doctor, or do you want to be on first name basis? What do you prefer?
I'm completely fine with Marina, Paul.
Okay, good. We'll be with first name basis. Okay, so I'm looking at the results right now, and I'm not sure whether you see those results or not, but I'm gonna. Okay.
Yeah.
So next question: the Paso Doble study of dolutegravir lamivudine versus bictegravir FTC TAF compared those two regimens in people with HIV who are virologically suppressed. Which of the following was demonstrated by the study? A, more virologic blips in the dolutegravir lamivudine arm. B, a mean of approximately 1 additional kilogram of weight gain in the bictegravir FTC TAF arm at week 48. C, an increased incidence of adverse effects in those who previously received nucleoside reverse transcriptor inhibitor-based therapy. Or D, virologic failure with integrase strand transfer inhibitors resistance in a small number of participants. So that's resistance to insties. Okay, we're going to vote now. And by the way, C should say who received non-nucleoside reverse transcriptase inhibitor-based therapy. I apologize for that typographical error, which is 100% my fault. Okay. And next is just my title slide. And once again, it's an honor to be here. I've got a picture here of the streetscape in Munich. Munich's a fascinating city. As you may know, these historic buildings had to be rebuilt after World War II because they were mostly destroyed, but they've done an extremely good job. And this is one of the more busier pedestrian thoroughways on a relatively quiet moment. I'm going to cover the prevention, treatment, and complications, but first a look back to when we last had the International AIDS Conference in Germany. So here's your question with a hint. When was the last time it was held in Germany, and where was it held? The hint is where it was held. Marina, you want to take this one on?
Oh, you want me to? Well, that is the Brandenburg Gates. I think we can all recognize that. It's pretty obvious. So must be Berlin.
That's correct. All right. We'll give you full credit. You don't have to get the year. Okay.
The year, yeah.
Okay. Anyway, it was 1993. 1993.
I wasn't there, by the way.
Yeah. So I'm old enough. I was there. It was an extremely depressing meeting. It was probably the low point in our early days of HIV therapy. We were using nucleoside monotherapy. It was obviously not working very well. And this is an archived piece from the Los Angeles Times, where it says the meeting ends with sobering appraisals, stresses prevention, not cure. It was also, I should just say, the time when we had the peak number of deaths of HIV in the United States. I'm sure this was true in Canada also. The early 1990s is when AIDS deaths became the leading cause of death among young adults in the United States. I had recently started my first full faculty position, and I had unfortunately experienced this phase. This is not a very happy phase, although it was still very gratifying, and our patients gave us a lot of hope. I will also show off here, and this is my very first research paper. This is one that I did under the guidance of my mentor, Dr. Marty Hirsch. You could see these very fancy poster that I put together, which involved printing 8.5 by 11 pages on my computer and then taping them to background red paper and then carrying that in a folder all the way to Berlin. All right, stop showing off. On to section 1, prevention. This was the big news of the conference. I'm going to just mention this study appeared at the same moment that Linda Gale Becker from South Africa presented it. Twice yearly lenacapavir or daily FTC TAF for HIV prevention in cisgender women. One of the things that drives me crazy, just quick parentheses, is that when you talk about treatment studies, you say TAF FTC, but when you talk about prevention, you say FTC TAF. And anyway, bothers me. And the same thing with TDF. But anyway, that's not the point. The whole point is to talk about this exciting study. Presented, published, same time. Here's the study design. Who's eligible? Who's eligible? Women in Africa considered to be at high risk for acquiring HIV who were HIV negative. And if they were HIV negative, they could then be randomized in a 2-to-1 fashion of experimental arms of lenacapavir injections every 6 months or oral TAF-FTC taken daily. So these are the experimental arms. You'll notice there's over 2,000 women in each arm. And then there's a standard of care arm, which is TDF-FTC, which we know does work as HIV prevention in women, and with a primary endpoint of HIV acquisition. You'll notice there's this other arm. These were women who screened for the study and ended up testing HIV positive. Using a technique known as recency assays, they were able to estimate the background incidence of HIV in the community, which led to 3 different comparisons: a lenacapavir versus background incidence comparison, a TAF-FTC versus background incidence comparison, and then also a TAF-FTC versus TDF-FTC comparison. Now, this was presented in July, but we had actually heard the news in June that the study was stopped. The Data Safety Monitoring Board said no need to go any further. The pre-specified efficacy criteria have been met, and then they announced the results, and that's what we get to see here. And here are those exciting results. This is from the published paper, but also presented at the meeting. And you'll notice that there are 0 infections in the lenacapavir arm, and that is obviously quite a bit lower than the other arms, either the background incidence estimated from the recency assays or the oral TDF-FTC or TAF-FTC. When Linda presented these results, there was spontaneous applause in the conference room. It was really extremely exciting. And she also got a standing ovation at the end of her presentation. Now, adverse events, of course, are different between these oral and injectable treatments. This was a placebo-controlled trial, so everyone got injections and everyone got pills or a pill. And it turns out that there were more injection site reactions in the lenacapavir arm. No surprise, these nodules we know are sometimes seen with lenacapavir, or actually always, because that's actually where the drug elutes out over the next several months. But you'll note that it was only 0.2% of study participants who had to stop. So let's take a look now at some of these comparisons. Lenacapavir, shown here, is significantly better than background incidence. So this is the first comparison. You don't need to be a statistician to see that the upper bound of this 95% confidence interval does not cross 1. By contrast, look at TAF-FTC. It does cross 1, so therefore the TAF-FTC is not better than the background incidence, but the lenacapavir is. So what about the next comparison? And that's compared with TDF-FTC. Again, lenacapavir is significantly better than TDF-FTC, whereas you'll see this big, wide confidence interval that overlaps 1. TAF-FTC and TDF-FTC are not meaningfully different. There have been people who have misinterpreted the study results because of the first comparison to say that TAF-FTC is no better, is actually worse than TDF-FTC, but that is not shown in this study. This study shows that the two are not meaningfully different. Well, what could possibly explain these results? What could possibly explain? Well, I'm sure you know that the history of preventive therapy in Africa has been checkered by some studies with very poor medication adherence to the oral regimen. And indeed, when you look at adherence over time, as measured by dried blood spots, you'll note that the adherence progressively worsens in both oral arms, so that by week 52, more than 80% in the TAF-FTC arm and more than 90% in the TDF-FTC arm are taking fewer than 2 doses of preventive therapy a week. There's only two numbers that could be: 1 dose a week or 0 doses a week. And we all know from previous studies that really a minimum of 4 doses a week for effective prevention for oral TDF-FTC. We don't know what it is for TAF-FTC, but it certainly doesn't look like it's 1 or 0 doses. A retrospective case control study looked at the cases of HIV that were acquired in the TAF-FTC arm. You'll notice that 92% of the people who acquired HIV actually had fewer than 2 doses a week. And this is significantly higher than the 66%, still not great in the women who did not acquire HIV. So what can we conclude? Hooray, twice year, injectable lenacapavir is 100% protective in women with high risk for HIV in Uganda and South Africa. Injection site reactions, though common, did not lead to study drug discontinuation very often. The adherence to both the oral regimens was low and worsened over time. There's a study called Purpose 2 that is fully enrolled, that is taking place in men who have sex with men and in trans women. And we understand the study results are likely to come out this year. I put 6 to 12 months just to be kind of like not overly optimistic. And then there are other studies planned, including a study I know in injection drug users. So this last issue, implementation, access, and pricing, they are already controversial. When I went to my first International AIDS Conference in Berlin in 1993, sorry, 1993, I actually saw lots of protests there, and similar to this study, this meeting, lots of protests already over lenacapavir. You might, wonder, what does lenacapavir cost? Lenacapavir is approved for treatment, at least in the United States. It's extremely expensive. I don't know what it's going to cost once it is approved for prevention. I don't think anyone does, but we hope that it's made accessible in the places that need it the most. That's going to be really critically important. Just Marina, do you want to comment on this study?
Yeah. I mean, obviously the study is fantastic for a number of reasons. I mean, it's really the first thing we've seen where we have 100% protection against in a prevention trial, right? We've come close before, but really this is phenomenal, particularly for women. So women have so far really had not benefited from most of the preventative therapies, and a large part of that seems to relate to the adherence issues, but maybe other factors as well. So one question for you that I wonder, do you think this is all about adherence? Do you think it's the formulation given every 6 months done by a health professional that ensures that there's coverage completely, or is there something particular, some special property of lenacapavir that is actually driving these results?
Great question. I've heard theorized that the fact that it has two mechanisms of action of antiviral activity might enhance its protective efficacy. Maybe, we'll see. The other is that I've also heard people say that perhaps it can be given once yearly because the half-life is so incredibly long. One thing I will say that's sort of adjunctly related to this, treatment failure with resistance to lenacapavir, it hasn't yet been reported. It might happen, but think about it. It's probably not of much consequence since we don't really use lenacapavir in any of our common regimens. It's obviously better not to have resistance, but you wouldn't harm someone by developing resistance to lenacapavir, at least given what their first-line regimen could then be. So
yeah, there are a couple of questions I think that follow from this that's come from the Q&A. Any data on self-administration of lenacapavir either in this trial or elsewhere that might help?
Not in this trial and not so far in the treatment space. There is an effort to make it a formulation that is potentially self-administratable, if that's a word.
Yeah, and I think just a final comment about the cost issues and the implementation. I think there was a presentation by Andrew Hill and his group. They've been very good at kind of taking companies to task for what's the true cost to develop these drugs and how could we actually arrive at a price. And so I think so much, and IAS has been really at the forefront of doing this, is a push to make sure that this drug is available at a cost that's affordable in the countries that really can benefit and need it.
Totally agree.
Yeah.
One thing I thought was worth doing was actually comparing the HBTN 084 study to the Purpose 1 trial for a variety of reasons. And I've put a title on this slide, which is What a Difference a Few Years Makes. So January of 2021 was one of the darkest moments during the COVID pandemic. It was the second time we had a major surge, and we still didn't have widely distributed vaccines. Whereas July 2024, there's still COVID, of course, but COVID is an entirely different beast. Both studies were stopped early by data safety monitoring boards. The HBTN 084 was presented first at a relatively small meeting called R4P, Research for Prevention, whereas this was given obviously a very important stage at the World AIDS meeting. Cabotegravir, superior to standard of care PrEP. You and I were talking about this, Marina, before the conference started. So that, has already been shown, and lenacapavir, same too. And then if you look carefully, if you really drill down on the treatment failures in the cabotegravir arm of HBTN 084, they were women who really were not adherent to the scheduled injections. So, the on-time injections, which are critically important for cabotegravir, are also 100% protective, just as they were in lenacapavir. We are talking about this because, we have available now cabotegravir for PrEP, but it accounts for a relatively small proportion of total number of PrEP users in the United States, at least. And I know in Canada too, since it was just approved there. I'm going to switch now to a different issue, which is presented by one of our distinguished graduates of our fellowship program. Right before the Munich AIDS meeting, there was a global, Microsoft disaster. CrowdStrike event happened. That is definitely a trivia question for the future. What event happened in July of 2024 that caused major traffic snafus and airline shutdowns? Delta Airlines is still recovering from it. Well, my colleague and friend and former fellow Rafi Landovitz's flight was out and out canceled. My flight was only delayed 2.5 hours. And he presented this virtually then from his home in Los Angeles. So as you may be aware, the guidelines for monitoring people on injectable PrEP not only ask you to get an HIV viral load with every injection, but they ask you to get it ahead of time. And so this is quite an ask. In fact, it's an impossible ask, as we'll come to momentarily. And the rationale behind that is that in 083, this is the study in MSM and trans women, what we had was a few people who actually had failure of cabotegravir despite on-time injections, and they ultimately developed integrase resistance. And what they did was they then went back retrospectively and said, well, if we had been using HIV RNA, would we have picked those up earlier? And the answer was yes. They would have been picked up earlier, and they might have been picked up before the development of integrase resistance. But the fact that they were showed that retrospectively doesn't mean that the testing strategy actually is implementable. Okay, that's another word, implementable. So what they did in this particular trial, which is the open label extension of 083, is that they evaluated whether if you prospectively do HIV viral loads on everybody, right, are you going to actually have decent test performance? And so this is the study design. At every visit, they got just a rapid test, an antigen antibody test, and a viral load. And then if any of those was positive, they would then have a very careful expert panel review the results and decide whether it's a true positive or false positive. And this allowed them to calculate the positive predictive value and false positive rate of an isolated positive HIV RNA. So the next slide is courtesy of Rafi Landovitz. You can probably tell this is not my slide design. This is his slide design. In this study, 26,528 viral loads were checked. That's a lot of viral loads. And in that group, since almost nobody acquired HIV, fortunately, only 73 had at least one positive test result. Among that 73, 27 had just a positive viral load. All right, so that's who we're focusing on. And when that expert panel got together and reviewed the results, they figured that 22 out of 27 were probably false positives and 5 were true positives. Additionally, they showed, and I'm not going to show you these data, that the viral loads in the 5 who were true positives were higher than the 22 who are the false positives, but not by a whole lot. They were sort of like in the 500 to 1,000 range versus like in the, 52 to 150 range. So not like a giant difference at all. So this led to a positive predictive value in the study overall of 18.5%. If you are a Bayesian, you won't be surprised to hear that if you had received your injections within the previous 6 months, the positive predictive value was only 9%. And when I say, are you a Bayesian, that means, are you a person who takes into consideration the pretest probability, the likelihood of a test being positive or negative ahead of time? And obviously, the people who are adherent to their PrEP are less likely to have a true positive than the people who are non-adherent to their PrEP. So if you look at this predictive value positive 18.5%, you might conclude that this is actually not such a great testing strategy, at least in some sense of the word. So to conclude, a positive predictive value of an isolated HIV RNA positive was poor, and really poor, if the person were taking their injections within the previous 6 months. They did note in the presentation, Arafi did note, that people who had positive results had all kinds of disruptions and chaos in their preventive treatment strategies as clinicians like you and me tried to sort things out. And then when they then repeated the people who had false positive results with another viral load, almost invariably it turned out to be negative again. So I think these results do call into question the guidelines recommended viral loads, and I wonder whether we'll see a change in those guidelines soon. Marina, can you tell me if in Canada they're recommending this as well for viral load monitoring for everybody on cabotegravir PrEP?
No, not at the moment. No, yeah, which is
Probably a good thing.
because
Yeah.
it's already, I think it's an important aspect about medicalization of PrEP and how actually much more monitoring needs to take place with PrEP, including, monitoring for other STIs. So it becomes almost, medicalizing the whole healthy person who's trying to prevent becoming infected with HIV. So it does complicate things a lot. Yeah.
I'm going to just show this poster very quickly. This is a community-based analysis from a database that that most people in the United States are not adhering to these recommendations. So you're supposed to get a viral load and a HIV antigen antibody test at every visit. And notice this last bullet, only 6% of the people in this community-based analysis actually did receive both tests. We are certainly not doing this in the guidelines recommended way. We are doing it every 4 months. We are using viral loads. So far, we haven't had any false positives, but I think we're going to see a change. I've now changed the title of this poster, saying need for revised guidelines for HIV testing in pre-exposure prophylaxis. All right, that was going to be the main studies of PrEP that I was going to discuss. Maybe this is a good time to talk, see if there are any other questions about PrEP before I move on.
I think the only questions that come up that we didn't really touch on have to do with women again and pregnancy. I think there was a question around safety of lenacapavir in pregnancy.
Well, one of the really encouraging things about this study design, which I thought was really quite brilliant, was that they strongly engaged with the community before starting the study and really asked them. They said, do you want to be able to continue treatment even if you are someone gets pregnant? And they said absolutely yes. And so this study did include quite a lot of pregnancies. Not surprisingly, when you look at the baseline characteristics, these are young women, they're sexually active, they actually had a fairly high incidence of bacterial STIs at baseline. So there were pregnancies that occurred, and there was no signal of any adverse events related to the lenacapavir. Good question.
Yeah.
So let me move on to this study. This is something called the PrEP-VAX study, PrEP-VAC, sorry. And this study is a study of both an AIDS vaccine strategy, 2 of them, DNA AIDS vax, and also an MVA, CMDR, MPLA vaccine or placebo, along with either TAF-FDC or TDF-FDC. So they again use this background incidence as the counterfactual, but there was a placebo arm for the vaccine strategies because, as you know, we don't really have an effective HIV vaccine. So I'm now going to show the results of the vaccine intervention and just walk you through this. This is the overall number of infections. This is the number in the placebo arm, and this is the number in the vaccine arm. And you might note that this 11 is bigger than 3. It's actually bigger than 3 by quite a bit. In fact, this met statistical significance for the vaccine being worse than the placebo arm. That's right, worse than the placebo arm. And this is not the first time that this has been observed in an AIDS vaccine study. And then the second vaccine, it wasn't quite as imbalanced, but still 2 infections in the placebo arm, 9 in the other vaccine arm. And this nearly met statistical significance. And I'm going to say this is another study that shows just how challenging developing an AIDS vaccine is. And I will also say that the additional efficacy of PrEP, as shown in the Purpose 1 study of lenacapavir, as shown in other studies of PrEP, mean that increasingly will be the standard of care for background HIV prevention. And being able to show an additional preventive effect with a vaccine on top of that is going to be all but impossible. Not only that, we have studies like this which show an increased risk of HIV acquisition. Now, the presenter threw in all kinds of caveats. He mentioned this is a very wide confidence interval, but nonetheless, the signal is definitely in the wrong direction. So I have this question, and this is one where you can answer: when will we have a safe, effective HIV vaccine that can be widely deployed? A, 5 years, B, 10 years, C, 20 years, or D, how about never? Okay, those are the choices, and you are going to be able to vote, and then we can ask Marina what she thinks in a moment, as soon as we see what the voters say.
This is definitely the question that I get most asked with my patients when they come to see me is, is there a vaccine? When's there going to be a vaccine? It's like, yeah. I think we've seen something as close to a vaccine as we're going to get for the moment, which is the lenacapavir data that you presented. It's not a vaccine, but it's, it's something that can be given once a year or twice a year to prevent HIV. We're getting close to something that's, as preventative as many of our vaccines we use for respiratory illnesses. I think this is, it's turned out to be such a challenge.
Yeah.
Such a challenge, and one would hope that with everything we've learned during COVID through mRNA technologies and vaccines, that we might have a step up and this could be advanced a little bit quicker. But this is clearly a very complex tasks, and this is disappointing, the results of the trial you just presented, and similar to some we've seen before, where sometimes the agent we're using together with, to generate the immune response is actually perhaps increasing risk, which is the absolute opposite of what we want to see, right? So I think we're beyond 10 years. I'm always an optimist, so I'm going to go with between 10 and 20.
All right, I'll go with 20 years too, but some of you said never. This allows me to share one of my favorite New Yorker cartoons. Bob Mankoff was the editor. He was the person who was like in charge of the New Yorker cartoons during its most brilliant phase, and he was a brilliant cartoonist himself. This is like the peak of social nihilism here, when you're trying to plan something with someone you don't want to see, and you say, "No, Thursday's out. How about never? Is never good for you?" Anyway, sorry about that depressing note, but I thought it was important at least to know about this study because it didn't get quite as much attention, obviously, as something that was positive, like the lenacapavir trial. All right, now on to treatment. So there were no important studies in people starting therapy at this entire conference. I looked, nothing. If someone saw one, please put it in the chat. And what is the explanation for this? Well, I'm going to take a metaphor that actually the first time I heard this metaphor used, it was Mike Sag. Mike Sag said, back when TDF-FDC Favirenz came out as a single pill, he said, it's like Godzilla. It's just stomping through all the other regimens because everyone was going on the 1 pill a day regimen. I think right now with our initial regimens of TLD globally, where over 20 million people are receiving it, in the United States, bictegravir, FTC, TAF, and to a somewhat lesser degree, dolutegravir, lamivudine, are just so effective and so easy to give that it's really a high bar to come up with an initial regimen that is going to be better. I am enthusiastically looking forward to the doravirine studies that are comparing it with bictegravir, FTC, TAF, because that will be an initial therapy study versus an integrase inhibitor, which we've never seen doravirine versus an integrase inhibitor. But there really has been hardly anything of note recently. There is another study. There's a study called LAPTOP, which is in people with advanced HIV disease, comparing boosted protease inhibitor of darunavir, cobicistat, FTC, TAF versus bictegravir, FTC, TAF. We'll see. We'll see. All right. So I didn't miss anything, Marina, did I?
No, you didn't miss anything. I think we'll see in a year or two some there's a lot of nice trials going on right now with like new formulations and longer acting injectables. So I think we may see stuff in the coming years, but you're right. Yeah, right now, no.
Okay. So what about switching? Well, lots of switch studies. This is a study of people on bictegravir, FTC, TAF, which is, as I said, the most commonly prescribed regimen in the United States right now. And they were randomized, if provided they didn't have Hep B or prior resistance or failure, to switch to dolutegravir, lamivudine, or to continue the bictegravir, FTC, TAF. It's not a huge study, but it's not tiny either. The virologic results are completely as expected in this group of patients who had never failed virologically. They continue to do really well, right? There was no emergent drug resistance in either study arm. There were more patients who discontinued due to adverse events in the dolutegravir, lamivudine arm. And, when there's an open label study like this, from a clinical trials perspective, you don't know whether that's because they really did have more adverse events or whether they basically, any symptom that they had after switching, they ascribed to their new medicines and it really wasn't drug related. One thing of note is that there was no difference in weight at week 48. There was a numerical difference, but not a significant difference. And this is something to keep in mind. I put this in a red rectangle, just so you'll remember this for later in this presentation, not in this study, but other studies. So that's a switch study in people with no resistance. What about switch studies in people with resistance? Well, we had an update on the study, which I had previously been calling D2F. But I asked the investigators, I said, well, how do you pronounce this D2F? And they said, DEFT. Everyone should know it's DEFT. Did you know it was DEFT?
No, I didn't.
Okay. Anyway, I guess if you learn nothing else at this webinar, you learn that D2F is pronounced DEFT. The D2 stands for dolutegravir and darunavir. And if you look at the way they write it, it's D superscript 2 EFT. And I guess superscripts are silent.
Silent.
Okay. You could have said D squared EFT. All right. And then there's another study called Visant and a third one, which was done at the Gaskio Clinical Research Site in Haiti. And all of them really give us reassuring information about using dolutegravir and bictegravir in people with nucleoside resistance. And they asked then as a supplement to the Nadia trial and the 2SD study and the Dawning study, but in particular, Nadia and 2SD. So let's look at DEFT. So DEFT was done in many, many countries. It was mostly in Africa and Asia. And they were basically people who had failed their first-line regimen with 2 nucleosides and a non-nuke, but had never been on a protease inhibitor and never been on an integrase inhibitor. And they did not have chronic hepatitis B. And the initial idea was to study the 2 new drug class regimen of dolutegravir and darunavir ritonavir. And that made a lot of sense because that study, that boosted protease inhibitor plus integrase strategy, had previously been studied with first older drugs, lopinavir, ritonavir, and raltegravir, but it wasn't so great for the obvious reasons. The lopinavir has a lot of side effects, and the raltegravir was twice daily, and it had, not resistance barrier isn't as high. So this was a good idea to study this regimen. And the standard of care was just to recycle the nucleosides, right? And use boosted darunavir. That's the WHO standard of care. Lo and behold, in the middle of this study, Nadia comes out and they modified the study so that this third arm of dolutegravir plus nucleosides, plus tenofovir nucleosides entered the fray. So then we ultimately have 3 arms, roughly equivalent size, over 200 randomized. It's an open label trial. This comment here, third arm was incorporated via a multi-arm, multi-stage design. This isn't so easy to do in the middle of a study, but they did it. I think it was actually made a lot of sense. So if you look at the baseline characteristics, 64% are women, 65% Black, 25% Asian, 10% other. CD4 at the time of switch, 200. Viral load at the time of switch, 20,000. And they had been on ART for about 5 years. There did not appear to be any meaningful differences between the 3 study arms when they were compared by baseline characteristics. So here are the results. On the left, proportion with viral loads less than 50. The biggest bars are the 2 new drug class regimens of dolutegravir plus darunavir ritonavir, and then also second biggest is dolutegravir plus tenofovir plus lamivudine or emtricitabine. Both of these arms are now at week 96 superior for viral loads less than 50 to the standard of care arm of darunavir plus nucleosides. So that is a news, because before, only this darunavir plus dolutegravir arm was significantly better. Now these 2 are better. I don't think they did a statistical analysis comparing the TDF, the dolutegravir versus darunavir arms, where both of the good dolutegravir arms, the one that gets darunavir and one that gets nucleosides. But that's an interesting observation. I'm going to say that the study presenter certainly thought that this supported the WHO new recommendations to use TLD, tenofovir, lamivudine, dolutegravir, in people who are on first-line regimens that have failed, consisting of a non-nuke and 2 nucleosides. Now you'll say, well, what was the resistance to the nucleosides in the people? But those data have not yet been presented. Now we're going to talk about a second study called VISAND. VISAND again looks at this group of people who are on efavirenz plus XTC plus TDF or nevirapine-based ART for more than 6 months. And there were 2 basic populations in this study. The first population was Arm A, and Arm E are considered virologically suppressed by WHO criteria, using a viral load of 1,000. That is a typical criteria used internationally. So that's Arm A, and it's really a comparison between dolutegravir plus TDF and dolutegravir plus TAF, and both of them get, of course, 3TC or FTC. So that one is a standard switch trial of virologically suppressed people. But there's also a failure arm, and this is a group of people whose viral loads is greater than 1,000, right? And they get randomized to dolutegravir plus nucleosides, TDF 3TC, TAF FTC, or standard of care. Now, standard of care for a long time was to use boosted lopinavir, which we've already criticized, or boosted atazanavir, plus zidovudine lamivudine. Now, why zidovudine lamivudine? Well, that's because their baseline nucleoside was tenofovir. And remember, high incidence of K65R in people who have subtype C virus and fail on a tenofovir-based regimen. So it made sense from a virologic standpoint to use zidovudine empirically if you don't have real-time genotyping. So let's look at the baseline characteristics for the VISAND trial. 60% women, median age 40. The suppressed arm, the viral load median was 84. Most were virologically suppressed, and the CD4 was nice and high, 491. By contrast, the viremic cohort of people who enrolled in the study had a viral load of 150,000 and the CD4 of 225, so much harder to treat that group. And in Arm B, 92% had nucleoside resistance, which consisted of over half with no predicted tenofovir activity, Most likely this means, K65R or SF70E or multiple TAMs, although I doubt that was it because they were on tenofovir. And 75% had no activity of 3TC or FTC. The boosted PI arm, which is going on zidovudine, 46% had zidovudine resistance. So the suppressed arm did really well. You can see that both the TAF FTC dolutegravir arm and the TDF FTC dolutegravir arm did well. That's perhaps not surprising, but you really see a difference when you look at the people who had viral loads who had failure. This is the failure group. And there's a separation here at the end of this week 144. This at the top is the dolutegravir strategies. Down here are the boosted protease inhibitor strategies, whether it's atazanavir or it's zidovudine. It's clearly not doing as well. And that's true also at the less than 50 criterion. Now, you remember in Nadia, there were some people who failed this dolutegravir strategy who developed integrase resistance, but not in Vizend. In Vizend, if you take a look, this is the number of people in Vizend who ended up having one or more major dolutegravir resistance mutations, and you can see that there were none in the dolutegravir arms. That's really encouraging. By contrast, there were 4 people in the protease inhibitor arm who did develop protease mutations. I am going to say that this is not real life. This is your clinical trials. There are people, obviously, in Africa who are developing dolutegravir resistance, and we're going to hear more and more about them. But whenever you hear about them, just consider the size of the treated population, which, as I mentioned, is in the tens of millions. So if we, not surprising, we're going to see some dolutegravir resistance, but at least the results of the Vizend trial, and I bet the results of the DEF trial are going to say that this is going to be an uncommon occurrence. So the last study I wanted to mention is this one, which was done at Geskio, and I was the presenter. You'll notice that this is not a picture of me. This is a picture of Dr. Samuel Pierre, who was supposed to be the presenter, but he had some difficulty obtaining his visa, so he couldn't come. And it's also not a picture of Serena Koenig, who's the senior author of this study and really the person who designed the trial, but she couldn't make it for a variety of reasons. So I was a co-investigator, and so I presented it on their behalf. This is a switch trial conducted in Haiti on people on second-line boosted PI regimens. You could see the study, the characteristics of the people in the study on the left. They were typical second-line treatment successes, people who had once been on a non-nuke and 2 nucleosides, now on a boosted PI, and suppressed. And they also couldn't have any active infections. And then they were then randomized to go on to oral bictegravir FTC TAF or to continue their boosted PI regimen. Now, this is the first study of its kind, specifically looking at this population. This is a population that is known from previous studies to have a very high incidence of underlying nucleoside resistance. By previous studies done in Haiti, this group usually has about 30 to 40% of people have tenofovir resistance, and a much higher proportion have 3TC or FTC resistance. So here are the week 48 results. They really are outstanding for both study arms, but in particular for switching to bictegravir FTC TAF. And you'll see that the study results, the upper bound of the 95% confidence interval is 0.2. So the non-inferiority margin was way up here at 4%. So switching was clearly non-inferior. This was, as I said, an open label trial. I also want to mention that it's amazing that they were able to do this study. This is a time of enormous controversy and civic chaos in Haiti, since Marina is in Montreal and I'm in Boston. So the big cities for immigration for people from Haiti are Montreal, New York, Boston, and Miami. And oh my goodness, things sound really terrible there. And Serena Koenig, who's the senior author, couldn't even visit the study site, which she had been to so many times, because actually there was nobody from the United States traveling to Haiti during this period except for family. But they were able to do the study and have very good medication adherence. So hats off to them. Anyway.
Really remarkable, yeah.
Yeah. So a couple of additional results from this trial. When you switch off a boosted protease inhibitor, you triglycerides drop, as they did here. There was more weight gain in the bictegravir arm than the dolutegravir arm, but I'm sorry, making in the bictegravir arm than in the boosted PI arm. A lot of the people in the boosted PI arm were actually on TDF as their nucleoside, which raises issues about, if you're going to see TDF nephrotoxicity, it's going to be on the boosted PI regimens. And indeed, one of the most common reasons for screen failure was that their estimated GFR was too low. We have collected resistance data, but they have not been analyzed yet. There's no lab in Haiti that can do HIV resistance genotyping, and there's certainly no lab that can do proviral DNA genotyping. So I think this study does support the safety of switching to bictegravir FTC TAF. We have other studies that have supported switching to dolutegravir-based regimens. This is really kind of the first one in a decent sample size to show that nucleoside resistance probably makes no difference for this switch. So now I'm going to ask this question, and then we'll also get Marina's view. It's going to be a hypothetical. It's about a patient with longstanding HIV and high-level resistance to NRTIs. K65R and 184V is currently virologically suppressed on a 3-pill-a-day regimen of darunavir, cobicistat, dolutegravir, and doravirine. They ask about switching to a 1-pill daily regimen. So what would you do? A, abacavir, lamivudine, dolutegravir. B, bictegravir, FTC, TAF. C, darunavir, cobicistat, FTC, TAF. Or D, I would not recommend switching. All right, 1-pill-a-day regimen for this person. I couldn't put in a dolutegravir option because that has lamivudine because it's a 184V patient. So I don't think we don't endorse that.
Well, I think this is like, I will wait for the results. I think it was the interesting things about all of these studies that you presented, and which really, I think, goes against our intuition about K65R it being such a deadly mutation for tenofovir, is that, we got these great results in those settings where there was a high prevalence of K65R. So are we seeing that same story repeat over that we see with the 184V, the replicative fitness hit that's taken with the virus when you continue to expose it to a drug that will keep the mutation around? And this Mark Wainberg, coming back to tell us,
yes,
he always said was true, and it's proving to be so in practice.
Yeah, the late Mark Wainberg, Montreal virology superstar.
Dear to my heart, Mark Wainberg. So, and I would say, and these are the cases where actually we do this often in consultation with our clinic pharmacists. We're very fortunate to have, and they used to be way more conservative, and we'd be kind of pushing the envelope a bit as physicians, and now they've become more, cavalier, and I'm always still like, oh, really? But I have done this in several patients that are similar to this with moving to a BIC FTC TAF, and have been astounded by first, the response rates, but also how much it makes a difference to them when they come off of these multi-drug regimens and they connect.
Well, I thank you, Marina. I think that this plus the 2SD study, these studies plus the 2SD study really do suggest it's safe to use these second-gen nucleosides, second-gen integrase inhibitors plus tenofovir XTC, and it works fine. On a related issue is this new strategy is basically to use an oral 1-pill daily of bictegravir and lenacapavir, although in this study, bictegravir and lenacapavir were given separately at 2 different doses versus their background regimen. And their background regimen was complex. It had to be complex. So what did this mean? It means that they were taking more than a couple of pills a day. They were taking multiple different drug classes. And the purpose of this study, this is the week 48 analysis, just to see, does this work? You'll notice the sample size is small. This is a phase 2 trial. And the switch generally did work. You'll notice that there's 92, 90, and 100% maintaining suppression. The safety profile seemed good. This is an open label trial. The results are felt to be strong enough that now there's a phase 3 trial using a co-formulated version of bictegravir and lenacapavir 1-pill daily for people with complex regimens, and we'll see the results of that study probably sometime next year. It will be competing against single pill regimens like bictegravir, FTC, TAF, and we'll just see if there are potentially some advantages of this new strategy. Okay, the next section is on complications. I want to make sure we have time for questions. This is the most important of the studies. This is the Paso Doble study. Now, Paso Doble is different from Dyad. Why is Paso Doble different? This is also a comparison of dolutegravir, lamivudine, and bictegravir, FTC, TAF, but the baseline regimens were not dolutegravir and not bictegravir. That's a key difference. Also, it's a fully powered trial. In fact, it's powered on efficacy and weight changes. And there are many, many other things they're exploring in this study. This is a study being done in multiple sites in Spain. I'm just going to cut quickly to the virologic results, which are completely unsurprising. This is a group that's never failed treatment before, has no known resistance, and switching to dolutegravir lamivudine is just as effective as staying on their other regimen. It's just as effective as bictegravir, FTC, TAF. We've seen this now before, that this dolutegravir lamivudine regimen is very effective in people who are virologically suppressed and have no resistance. This is Tango and Salsa, played out in a different population, slightly different population. Okay. They did do a blip analysis, and they said blips were not more common in the dolutegravir lamivudine arm. In fact, numerically, they're more common in the bictegravir arm. But this is the result that got the most attention, and I took this from Esteban Martinez. This is the changes in weight over time, that there was a nearly a two pound or 1 kilogram, nearly a 1 kilogram more weight gain in the bictegravir FTC TAF arm than in the dolutegravir lamivudine arm. And we all know that the weight changes after switching depend a lot on what you were on previously. And in this study, it was really interesting in ways that we don't quite understand. If the person were already taking a TAF-based regimen, there was no difference in weight. If a person was not taking TAF or abacavir or TDF, there was no difference. So if they're already taking a 2-drug regimen, like a boosted PI plus lamivudine, then they didn't have any changes in weight. Or so the difference really was made up mostly in people switching off abacavir and in particular TDF. But the question then is, why wasn't that seen in the dolutegravir lamivudine arm? And I don't really understand that, because certainly in the Salsa trial, people on baseline TDF did gain more weight when they switched to dolutegravir lamivudine, but that was not seen in this trial. It was a little bit different because that was a comparison of staying on TDF versus switching to dolutegravir lamivudine. So not sure. So as I said, virologic results as we expect. Weight changes are surprising, at least to me. And when I contrast what we saw in Tango and Salsa, a little different. And also there's an observational study from Spain that does not show a difference in weight trajectories between these two regimens. We're not sure why the weight gain differs based on baseline characteristics. One question I had for Esteban at the conference was, was there a imbalance in randomization, that if you had more people in the bictegravir arm who were baseline regimen was a TDF, FTC, efavirenz, that could have driven the result. But he didn't actually know. There did seem numerically to be more TDF in the bictegravir arm, but I'm not sure. Do you know what is your hypothesis for this?
Yeah. I mean, I think it's starting to be appreciated that TDF has a weight suppressive effect, right?
Yep.
And coming off TDF, you'll see this larger gain of weight, and same as you mentioned with the efavirenz. So but that doesn't explain then why you didn't see that with the.
It could be just that you get more weight gain with bictegravir TAF, but it's funny because this there is this 19% versus 40%.
That's huge difference.
Yeah.
I mean, there's still relatively small numbers, but still large enough to think that there's something there. And I guess the TAF, this interaction between TAF and bictegravir that we sort of had a hint of in previous studies may be playing a role.
And then this one I don't understand at all. Like, if your baseline doesn't have TAF or abacavir or TDF,
Yeah.
And
No,
there's no difference at
change.
all. So,
Yeah.
okay, well, we haven't figured it out. Okay.
Another weight
Um,
mystery.
yeah. So I did want to bring up an old story, which is a back of ear and cardiovascular disease. It's not really that important any longer for most of us who are not using much of back of ear at all. But there are this sort of legacy of patients on a back of ear, some of whom are getting to be quite old. And I think that if anything, this study might get people to think, look, if dolutegravir lamivudine works so well, why do you need to be on a back of ear dolutegravir lamivudine? So that's an important message. So this is the REPRIEVE trial. And remember, the REPRIEVE trial had independent adjudication of cardiovascular events. So even though it was a study of pitavastatin versus placebo, using it to investigate the sabacavir exposure as potential risk factor for cardiovascular events is stronger in this study than it is in most observational trials, because this is in the context of a prospective clinical trial with independent adjudication of events. So what did they find? Well, it was significantly associated with cardiovascular events when controlling for baseline characteristics. This was not observed for tenofovir, PIs, or thymidine analogs. I do want to say that that I've not seen before that former use of abacavir was associated with excess cardiovascular events. There was this hypothesis that there was this sort of switch on and off that might have caused this. I'm not sure. I will say, again, I'm going to emphasize this, there doesn't seem to be much need for abacavir any longer. But, every time I see prescribing data, it's like there's still some people who are on abacavir. We had a patient admitted to our hospital recently, a man in his late 60s. Even if he doesn't have cardiovascular risk factors, there's probably no need for him to be on abacavir anymore. The leading cause of death in men in their late 60s in the United States is heart disease. So,
Yeah.
what's Canada like these days for abacavir
Yeah,
use?
well, we were very big users of abacavir in Canada, so it's probably been even worse, slow to switch off. But there's been a big movement to switch off and to when you can to use 3TC dolutegravir
Yeah,
regimens. And just remember, Reprieve, I mean, this is such a great trial because it's just going to keep giving. I mean, there's so many
yeah,
great lessons from it. But remember that the people who are enrolled in Reprieve were actually low risk to maximum moderate risk cardiovascular disease. So
very good.
that we're
Yeah,
actually seeing this
yeah,
signal this strong, and the former as well as current users, I think is really an opportunity each time you go to discuss, starting a statin in somebody, you should also revise their
yeah,
antiretrovirals and they're not
yeah.
on abacavir.
Couldn't agree more.
Yeah.
I'm going to do one more complication trial because it occasionally comes up in clinic, and that's insomnia for dolutegravir regimens. And this is a trial that they intended to enroll at least twice as many people, but because of the pandemic and other issues, that they could only enroll 19. And what they took were people on dolutegravir regimens who had insomnia. And if you recall back to the early clinical trials of dolutegravir, the single trial, which was compared dolutegravir, abacavir lamivudine with TDF, FTC, efavirenz, there was actually more sleep disturbance in the dolutegravir arm, which was surprising. So they then switched them, half of them to, or about half, to bictegravir, FTC, TAF, and continue the rest on dolutegravir. And the study was open label. And for what it's worth, the subjective sleep quality improved, which is good, although it might just be suggested by the open label nature design. But what they showed was some very fancy functional MRI images that suggested that this was associated with an improvement in what's called functional connectivity. And functional connectivity is good for sleep. And so they also measured biomarkers, which of course didn't show anything because they never do. So anyway, just something to consider. It could be that abacavir is partially responsible for insomnia because some of the people on dolutegravir in this trial were also on abacavir. This, my last section, is going to be on this, which is a cure. Hooray! Hooray. So started with Berlin, ending with Berlin. This is a person in Berlin who was, quote, cured, and we'll see the details momentarily. These are the previous cures, and notice they all received CCR5, delta 32, so homozygous donors, allogeneic hematopoietic stem cell transplants, and they received them for underlying cancers. And several of these people have been generous enough to come to be public about the fact that they were the cured person. In addition, there's one person who seems maybe to be cured in Geneva with just a wild type donor. And there are also a bunch of failures. We had two of these failures in Boston, very notable. When they failed their cure, they actually had massive viremia rebound, lots of susceptible target cells and acute HIV all over again. But this patient is in Berlin. He born in 1964, diagnosed in 2009, had CCR5 wild type and delta 32. So he was a heterozygous himself. So he had one delta 32 gene and one wild type gene. And then he gets started on ART around the same time he was diagnosed with leukemia. And his donor is a heterozygous. They tried to find a homozygous delta 32 donor, but it was just the heterozygous donor. And then, as is often the case, there's a full chimerism achieved. And I got to hand it to them. They stopped the treatment in 2018. The patient requested it, and they stopped. And when I say I got to hand it to them, they did not report this for over 5 years. Because I think, all of these cases, people are just waiting for that sort of random moment when something in the reservoir pops up and, replication starts again. So they showed, all these different sanctuary sites. They biopsied. They found no evidence of virus, plus the waning HIV-specific antibody and T cell responses, and so they're not exposed to any HIV antigens. What the presenter suggested, and I have to say he did a super job presenting this case, very detailed case presentation, strongly suspects that it's the donor cells that are providing some anti-HIV activity, alloreactivity, and that's been a leading theory. All of the patients with successful cures with this strategy have had some degree of graft versus host disease. So anyway, another cure. Hooray. Not widely deployable. No. Okay. All right. Should we do these post-test questions?
Sure.
All right. We can do the post-test questions. Yeah. Anyway.
And then we can have some time
Have
at
some
the end
more
for
Q&A.
wrap-up questions, Q&A.
So
Yeah.
way back to the beginning of this presentation, study of lenacapavir and tenofovir alafenamide, emtricitabine for PrEP in women. Which of the following was observed? Breakthrough infections leading to resistance to lenacapavir, superiority of TAF-FTC over background incidence, progressively worse adherence over time with TAF-FTC associated with risk of acquiring HIV, or higher incidence of HIV in the TAF-FTC group than the tenofovir diazoxyl emtricitabine group, significantly higher. So which one do you think it is? Okay. That adherence issue is a... All right.
All right. It went up.
Well done, group. That seems to be, I actually heard someone say at the conference, in settings where you think the medication adherence is going to be poor, you should strongly push injectable PrEP. And, you see results like this and you really have to.
I think it was interesting that the design of the study was actually to look with the theory that TAF-FTC would have better adherence because it's a smaller pill, and that was the rationale for actually testing the two, whether that makes real sense or not. It did sort of have slightly better adherence initially, but that waned just the same way as the TDF-FTC, and it just, I think, especially in this population, it's really hard.
I think it was 84% versus 92%
by
the end of the study.
By the end it was.
All right. The correct answer is adherence to both the oral regimens decreased over the course of the study and was associated with risk of acquiring HIV. TAF-FTC and TDF-FTC were not meaningfully different. They both were pretty ineffective, actually. Lenacapavir was clearly the winner. All right, Paso Doble compared bictegravir FTC TAF with dolutegravir lamivudine in people who are suppressed. Which of the following was demonstrated in the study? More blips in dolutegravir lamivudine, a mean of 1 additional kilogram of weight in the bictegravir FTC TAF arm, increase in the incidence of adverse effects in those previously receiving non-nucleoside reverse transcriptase inhibitor-based therapy, or virologic failure with integrase inhibitors with resistance. Okay, go ahead and vote. We can't vote. All right.
Good. That went up. Yeah.
Yeah. Great.
Great. Okay. I guess we have a few minutes, and so we might as well, there's a few questions that we didn't get to in the Q&A, and maybe just group them. A lot of them sort of circle around lenacapavir and some of the different aspects we may use it. Oh, you have your finger up. Yeah.
Yeah. Could I just thank, there are a bunch of people I wanted to thank, and I didn't get a
Oh,
chance to go to my
absolutely,
thank
yeah.
you slide.
Sorry.
That's okay. I want to thank Serena Koenig, Samuel Pierre, Rafi Landovitz, Esteban Martinez, and I want to thank you, Marina, for moderating. So,
Oh, no, my
all right.
pleasure. It's been fun. Yeah, so I guess one of the questions, I think it was actually asked during the presentation, but some participant has asked about the Levi syndrome, or whether it's possible that people who become infected while receiving lenacapavir might have a delayed detection of that infection and ultimately have a resistant virus that they're infected with. And do we have any evidence of that potential? Or are people looking for it? Do we know anything about that?
They're going to look back at the samples and see if they have any positive HIV viral loads, but so far, no evidence of that. It's certainly possible. I do want to say that those few breakthrough cases in HPTN 083, and I think there's been at least, I know there's been at least one in clinical practice,
Yeah.
but it's uncommon.
Yeah.
People who are on time with their cabotegravir injections don't have a lot of failures.
Yeah.
And we certainly haven't seen one, and I only know one case it was published. So this is really going to be rare.
Yeah.
Granted, it's worth worrying about, and I think the question was a good one about lenacapavir, but we don't know yet.
I think there's also quite a few questions about where is lenacapavir going to fit in, our treatment armamentarium going forward, not for prevention so much, but in the treatment side with Biclen and the costs, and how are we going to, where are we going to use it?
If you ask me, the most interesting aspect of lenacapavir for treatment is likely to be the once weekly strategies, because it sort of falls in between this every 6 months for PrEP and the daily pill that is being developed, and is something that, at least based on surveys of patients, they'd like to be able to do. Take a pill once a week. Now, the one that's ongoing is with islatravir.
Yeah.
But it also is being looked at with other drugs. So
good. I think that wraps things up. There was a question, I guess, on the PrEP side about what do you do in practice if you're monitoring someone with RNA testing on PrEP and you get a positive HIV RNA results? How would you manage that?
Such a great question. Every single reactive test in patients on PrEP, based on my experience, is extremely difficult to adjudicate. We contact it, whether it's injectable PrEP or oral PrEP. First, we contact the pharmacy and find, have they been filling it regularly? because often you will find that they have not been filling it regularly. And indeed, that's a reactive test that needs to be carefully attended to because it might be a true positive. If the person's actually adherent to PrEP and they get a positive viral load, I would encourage you to discuss with your patient that you cannot tell if the viral load is very low, whether it's a true positive or false positive, and then discuss with them the options of either first repeat it, because if repeating it would be the first step. But then if the second one comes back also low level positive, discuss with the options of stopping and then monitoring, B, continuing PrEP and seeing if there's evolution of their antibody result, or C, if they really think that they're at high risk and they want to go on suppressive ART, going on suppressive ART. I know that some people want to order an HIV DNA test, but there's no actual good data that I'm aware of that that's more sensitive than a standard HIV RNA. I mean, I think they have the same sampling error problem that the DNA genotypes do. What do you think?
Yeah, no, I agree. It's yeah, and I think you may just get into, I mean, the false positivity problem with these, you know, it's not risk population, but the prevalence of a true positive in that population is going to be extraordinarily low because
Right.
they're adherent to the PrEP.
My most recent example is that the person who showed up in the emergency room diagnosed with syphilis, and his HIV antigen antibody test was positive, and then his confirmatory test was negative, and he said he was taking his PrEP religiously, but not according to any of his refill records.
Right. That's so unfortunate. You have to do that detective work.
You know, and then the question is, had he resumed taking his his PrEP after starting to have more at-risk behavior? And that's why he's now partially suppressed. And so I told him, I said, the only way we're going to be able to sort this out is if you stop your PrEP and we follow you longitudinally. And he said, okay, so we'll see. I don't know what the result's going to be.
Oh boy. Yeah, those are tricky, really tricky. I guess I think we're going to have to wrap things up. It's too much fun, but we have a few closing slides. First of all, I want to just thank you, Paul, as always, brilliant and really entertaining and interactive. And it's been such a pleasure. I hope we can do this again. Please provide your evaluations to us, and there'll be information there on how to claim your CME. This feedback is really important so that we can go forward. There's more coming. Don't forget to keep an eye out for these new other webinars coming. So the next one along COVID. And here's some other ones that were coming up soon. More on complications, on screening and prevention of anal cancer. I think there was some of that stuff presented at IAS, but very kind of complicated algorithms to do so. So we can hear about that. Opportunistic infections, which are still with us, unfortunately. Of course, don't forget CROI 2025 in beautiful San Francisco next March. And thank you again, everybody. We had a tremendous turnout today, and really glad to have been here with you, Paul, to do this webinar.
Thanks, Marina.
Yeah, bye-bye.